When people think about fungi, they mostly think about the mushrooms they eat in their dishes. And when people think about bacteria, they mostly think about their diseases and illness. But both fungi and bacteria are very common inhabitants of our soils, where they play a lot of roles, including the favoring of plant growth and also activities in the degradation of organic matter. Now here we are where we have an archaeological site, and in this site the fungal and bacterial activity is important to explain what we'll find in the soil. For example, fungi and bacteria can degrade the organic matter, which explain to us the relationships between the different layers of the excavation, and they can also explain why we can find some organic remains as materials made by man in vegetal or animal materials. On the basis of the material available for fungal nutrition, which is in the soil, we can find different species of fungi, different communities. So the presence of some species of some kinds of fungi can explain the ancient occurrence of fires, of deposit of dejections, or the occurrence of a rich presence of food. And thus, if we characterize the microbial occurrence in the soil, we can have ideas about what was the use of the human use of a certain place in the past. One of the difficulties that archaeologists have here is to recognize what is the original hill natural layer and what is the layer of the mound. Now, if we recognized a layer in the depth where we have fungi, indicators of a rich presence of organic matter in the past, we could recognize that layer of the original hill layer, which fungi in the past degrade, but which are still recognizable now because fungi are very long-lasting organisms which can also resist in quiescent forms. Because of their deterioration activity, fungi and also bacteria are also a treat for the conservation of the findings which archaeologists obtain from their excavation. And it is very important to characterize what microorganisms are growing on the remains when archaeologists take them and bring them to the lab or do an exposition. Because if you do not take care of delete the microorganisms from the remains, there is the risk that they eat them and finally you do not preserve them for the future generations. In these days at the Tobiotsuka Kofun we collected samples brought from the outside trenches where we'll study the occurrence of the layer indicator of the bound between the mound and the original hill, and also from inside the stone chamber where we'll take care of the presence of fungi as potential deterioration agents of the remains which have been found. And in particular in these days we characterize some interesting biodeterioration effects of fungi on the potteries they already found. We collected many samples. Now a part of them is under processing here at the University of Okayama to prepare aliquots which will undergo molecular analysis to characterize the fungal diversity. Another part will be the object of archaeobotanical studies to find if some plant remains like seeds and fruits could be still found after hundreds of burial environment. And a part of these samples will be also brought to Torino to calibrate the techniques which we'll use on these samples in the next session of work here in Japan.